Telomeres. Whether you pronounce it telomeres or telomeres, what's important is how they function. Telomeres are non-coding DNA sequences at the ends of our chromosomes. But what happens is every time a cell replicates, or every time a DNA strand replicates, you lose genes. You lose those DNA sequences off from replications off of the chromosome. What the telomere does, it actually cushions that and allows the cell or the chromosome to replicate without shortening and without losing any DNA. And it does that because it has that repeating TTA-GGG DNA sequence, and it's on the ends of the chromosome, and it's up there about, sometimes between 100 to 1,000 times, or sometimes even more, as we've discussed in the last segment. So here we have an example of that chromosome. We give you a visual of the telomere and a visual of the chromosome and how it's going to look. Next, we're going to go ahead and discuss kind of how those telomeres actually cushion the cell and actually keep the chromosome from shortening.
Telomeres play a central role in studying fate and aging by adjusting the cellular response for stress and growth stimulation on the basis of previous cell division and DNA damage. Three major purposes of telomeres are: they help organize 46 chromosomes and nucleus, they protect the ends of our chromosome, and they allow chromosomes to be replicated properly during cellular division.
As you can see in the green, this is a chromosome, and this is a telomere. They help organize 46 chromosomes in a nucleus, and they protect the ends of our chromosomes. Also, they allow the chromosomes to be replicated properly during the cell division. As you can see in the photo, it caps off the end of our chromosomes, which DNA repeats over and over again with 3,000 through 15,000 in length.
In 1956, Francis Crick came up with the central dogma, which is DNA into RNA, which translates into a protein.
Transcription and translation. These are two processes that are used to create proteins. But transcription and translation are very similar to things we do every day. Transcription is like typing on a keyboard. Translation is the interpretation of what's typed on the keyboard. So let's give you an example. When we're texting today, you can text WYA, IDK, or WYM. And these sets of letters have meanings. Now we're going to further explain to you how these letters have relations to translation and transcription. So we have three letters. These three letters individually have very little or no meaning. But when you put them together, you take the three letters WYA. That can translate when you're texting into where you at. IDK translates into I don't know. And WYM is what you mean. Now, we're taking these three statements and we actually can create a sentence out of taking these letters, going from there to here, and then we actually can have a kind of a broken sentence. Where you at? I don't know what you mean. And that's our statement. And that's the same way that codons translate into proteins. So now we can use that same theory or that same technique when we're talking about translation of amino acids and translation of nucleotides. Each group of three nucleotides are a codon. So a codon is, again, a group of nucleotides that basically translate into an amino acid. So you have UAG translates here, AAG translates there, and UUU translates here, and there's also UAA, that's a stop codon. So that's the translation for that. It's stop. So there's no amino acid for that. So then we take these amino acids when grouped together, they're gonna come together and create a protein. The same way our letters over here translate into words and then translate into a sentence. Again, the translation is here from nucleotides to codons to amino acids to proteins. Let's say we look at, when we're looking at recombinant DNA technology, we're basically looking at replication. So if we take an E. coli bacteria, which is what they use, and they use that because the bacteria self-replicates. It'll make exact copies of itself. So if this square is a bacteria and it replicates itself, it's gonna replicate another square because that's what the genetic code has decided. When it replicates an exact copy of itself. It gives us another square. Let's say we take our square away and we wanted to replicate a rectangle. How will we take a square and get it to replicate a rectangle? We're going to show you right now. So let's take our rectangle away and we'll show you how it's done. The way it's done is you're going to use what's known as a restriction enzyme and you're going to use ligase. For our restriction enzyme, it's just like a pair of scissors. So what the restriction enzyme does, there are specified sites on the code of DNA and it cuts. It's a cutting enzyme. So it actually cuts out a certain part of the enzyme that makes this a square. So it cuts that, you're gonna pull that out. Then you're gonna recode that with a specific enzyme for a rectangle. So when we do that, you're gonna actually have that replace the DNA with this segment that's gonna make a rectangle. Ligase, that's like glue or scotch tape. So we're gonna take ligase when it comes back down with those genes, you're gonna take the ligase and you're actually gonna allow that to stick together. And then you're gonna put this, which will be like a plasmid, you're gonna put that back into the bacteria, just like we do on our printing machine, and it's gonna give us copies of itself, which is another copy, an exact replica of a rectangle. And I know that's a little oversimplified, but that's basically recombinant DNA technology. This should be it. Take it off the team. When we're talking about replication and you're talking about telomeres, the way they actually help the DNA strands extend their life and not be chopped down in terms of chromosome size is, let's say this is a replication strand. This is our 3 to 5 prime strand. Knowing that replication from DNA polymerase is only 5 to 3, this presents a problem. So what we have on this strand, we have replication taking place. We're going to have our Okazaki fragment come in here, and we're going to have our DNA polymerase come in here, and nucleotides are laid and replication takes place. You see that? So we're going to do that again. We're going to have a Okazaki fragment come in there. We're going to have our DNA polymerase come in here, and we're going to have also, we're going to add some more DNA to the strand. But what happens is now we have an end replication problem, because here at the end, you still have nucleotides that are unpaired, and this is how you get nucleotide loss. So that creates a problem. Now, the way telomeres solve that problem is because you have an actual enzyme called telomerase. Telomerase actually breaks the central dogma, because it actually has its own protein. It's a protein and it's an RNA. So it has its own RNA sequence on the inside of it. So what happens here is that when that when that telomerase comes in, that telomerase can actually lay DNA code sequences and lengthen the telomere so that some more replication can take place. Now, what you have happening here, once that's done, you still have an end replication problem, which you're always going to have. But because we've had the telomerase, it's adding on to the life of the telomere and lengthening the entire chromosome. When we're talking about DNA fingerprinting, we have to always keep in mind the term PCR. That's polymerase chain reaction. Polymerase chain reaction is the duplication of that DNA strain that you're trying to identify or compare with other strains. Once you have that, now you're talking about something called electrophoresis. And what that is using little electrodes to basically separate each strand of DNA from each other by length. What you need to do there is you need the agarose gel, you need the buffering solution, you need a comb and a little mold for the agarose gel once it settles, and you also need the DNA samples with pipers as well. Once you have that, you heat the gel up to loosen it, you pour it into the mold with the comb in it, and once that's done, you heat it up in the mold and then you let it settle. So you let it cool off. Once it cools off, you're going to use your pipers to get the DNA samples and put them into each one of those little small cells that you made with that comb in the gel. After that, you put the mold in the buffering solution. You add your DNA samples into each one of those little small holes provided by the comb. Then you turn on your electrophoresis device. That provides the electric current. What happens then is that the electric current starts to pull the pieces of DNA toward one pole. The smaller the DNA fragment, the faster it travels. The larger the DNA fragment, the slower it travels. And that's how you get all those separations on your DNA fingerprinting sheet. That way you can compare one sample with the next. And that's how you find out which DNA is a match.